if you're thinking this is a bit of a deja vu, you are right. Because almost exactly 3 years ago, I was standing in this room giving a presentation about the European lead factory. And here I am again 3 years later, doing the same thing. But it's not the same talk. So let me do a bit of explaining for what's happened there. So at the time, I was working at ChemAxon. I was leading the consultancy group, and this is one of the applications we built. 3 years ago, the system was going into production. We were putting it together, and we were telling you all about the system. Now the project has been running for 3 years and is drawing to a conclusion. The project terminates at the end of this year, and it's been a big success story. So ChemAxon invited me back to talk about it. In the meantime, because the project was already running, it wasn't really a new project, I retained the responsibility for basically managing it and keeping it going, so hence why it's me standing here doing this. So first I'm going to talk a little bit about what the European Lead Factory is. So it's part of the IMI project, the Innovative Medicines Initiative. And it's a project that was set up really to identify novel lead compounds that could be obtained from screening. And there are three categories of partners. There's the FPO members, that's the pharmaceutical companies in Europe. There are a number of SMEs, which are largely led by CROs who are doing chemical synthesis. And there are a number of academic parties who were also partly involved with the chemistry side and partly with the biology side. So, in essence, this is what the idea of the project was. There were two arms to the project: the chemistry side and the biology side. The chemistry side wanted to put together a set of novel compounds known as the Joint European Compound Collection, JECL. And that proceeded from 300,000 compounds from the pharmaceutical companies who trawled their internal collections and donated them to the project. And there are novelty checks and quality checks on there just to make sure that the good quality compounds were going on. This wasn't just a case of them getting rid of all the dross from their collections. They were trying to put high quality compounds into the set. They didn't want to just live with 300,000 compounds that already existed. They wanted more novelty. And so they wanted to synthesize around 200,000 compounds from newly synthesized novel chemical libraries. On the biology side, other partners were putting together novel assays that could be formed into screens. And then by combining the novel compounds and the novel assays in screens, you would hopefully end up with novel leads. The programme consisted of 13 work packages, of which ChemAxon was involved with 2 of them: the sourcing of the library proposals and the review and the selection of those library proposals. And so this is the workflow for the library generation. Basically, the library proposals were sourced primarily from the consortium partners, that's the CROs and the academics that I mentioned earlier, but there's also a crowdsourcing aspect. So anyone could come in and make a proposal to the library, and if the library was successfully synthesized, they would be rewarded. There was a cash prize for submitting a library. Those library proposals were put into the system and were assessed by the Library Selection Committee, and they're assessed on a number of properties on the right-hand side. To assess whether that library was suitable for synthesis. If it was approved, it then went on to experimental validation and design optimization, and eventually, if all things went well, the library was produced and entered into the Joint European Compound Collection. So there were a number of workflows involved, depending on what type of user you are, whether you were from a ELF partner or whether you were from an external party who is part of the EU or outside the EU. So this actual workflow for proposing the libraries and the process of assessing them was slightly different. The overall process was obviously the same because you ended up with a library that could be synthesized, but the individual workflow was a bit different. And the workflow itself looked like this. Firstly, the user signed up and registered themselves into the system. Then they entered details of the library into the system. And you basically specified a scaffold, and we used Marvin.js for that, obviously. And then there were 3 ways to actually upload the library into the system. Firstly, you could generate it externally yourself in whatever tools you wanted and upload an SD file that contained the library definition. Secondly, we could use the Marcouche library definition specified in MRV format that would be enumerated within the system to generate the library. And thirdly, for people who really didn't have the technical capabilities or the software system to do either of those, they wanted a really simple way where you can just sketch a library, pick from a predefined set of R groups, and let the system enumerate a sample library from that. So we ended up with 3 different ways of entering the library. You basically put in the details as was required there and entered it. Once that library was entered in, a set of initial property calculations was performed using the ChemAxon property calculators, fairly standard drug-like filters, log P, molecular weight, polar surface areas, fraction of sp3, hybridized carbons, etc., etc. In addition, there were some novelty checks done. So the enumerated library was checked against a reference set of 12 million structures that contained patented compounds from SureChem and from e-molecules to check whether those molecules were already commercially available or had been described in patents. There was also a search against the structures that had already been deposited into the Joint European Compound Collection. That was an external search that was performed by another party in the consortium because that information was proprietary. We didn't have access to the structural information, so that was called as an external service. And just to be safe, we run checks against the UK section of the compliance checker database, just to make sure there weren't any naughty compounds getting into the system that way. Once you're all happy with that set of compounds and the profile of the properties, you then submitted it into the system. And at that point, the library then became available for consideration by the Library Selection Committee. And at this stage, the user who did it has basically handed over ownership of that library to the lead factory. On submission, another set of property calculations is done. These ones were more expensive, so were delayed until the library had actually been submitted. And this does more sort of novelty checks. So we're doing fingerprint comparisons, similarity search, basically using ECFP4 fingerprints and a fuzzy pharmacophore fingerprint against the same reference set of 12 million structures. Again, to try to identify the novelty of the library. And this, this step was actually the birth of the MadFast similarity search. It was this process that actually was the driving factor that needed the MadFast to be created. Prior to that, ChemAxon did have some old molecular descriptor tables that did the job, they did the similarity search, but they were not optimized for performance. And for this sort of scale of operation, of comparing several hundred structures with 12 million structures or more, it would have taken days to have run that sort of search using the old technology. And hence the MadFast came in and was created. And that actually was able, at the time, I'm not quite sure what the current status is, but at the time we could do sort of 1 million versus 1 million searches in about 25 minutes. More than fast enough for what we needed here. Finally, well, not quite finally, but the next step here was the library would be assessed. Obviously, being an EU project, there had to be a committee involved, and that's the Library Selection Committee. So this is a bunch of old and wise people. They know a good library when they see one. They sat around a table or a virtual table and assessed the library based on all the properties. The properties provide some sort of level of subjectivity here. Objectivity here. So the libraries were being assessed by the same criteria, and so we're getting a fair and transparent assessment of the libraries. It wasn't really known, it was designed to be a sort of a transparent process. And the library was then scored against a number of criteria, and either it was approved, in which case it went on for experimental validation, or it was decided to be no use, so it was rejected, or there was a halfway house. It was considered a useful library, but not quite there yet. So it was basically put back to a refined status where it could be, the library design could be improved and then resubmitted for proposal, for assessment again. And then once the library is approved, it goes on for chemistry validation. The library might be synthesized after that if it's successful. The compounds will be added to the Joint European Compound Collection. and ultimately those compounds would be used in screening. So that was the basis of what I described 3 years ago on what had been set up. So what's happened actually over these 3 years? What is the outcome of the project? Well, as I said, it is very much of a success story. Here's a quotation from Professor Adam Nelson, who is the chair of the Library Section Committee, who was the guy who was overall responsibility of that. I won't read it out. I'll leave you to read it. And it's highlighted in a press release that's linked at the bottom if anyone wants to see the full one. But essentially what he's saying here is that the library, it would simply not have been possible to assess this number of libraries if we hadn't had a good tool for being able to submit the proposals and to assess them. And the bottom line is, well, the last sentence, the web portal developed by ChemAxon played a key role in this achievement. So a few details of what happened in the years of this project. Well, we had 92 users who registered to the system. 75 of those successfully submitted a proposal. Some registered but never got over the hurdle of actually submitting a proposal. That was understandable and expected. Of those 75 people, they managed to propose to submit 851 libraries for consideration. Of those, 61 were approved, 37 were rejected. It doesn't quite add up to 100 because there's still a few in process. 192 of those libraries have been synthesized. 77 at the last count were ongoing. They were in the process of being synthesized. And at the time this was assessed, which was a few weeks ago, 145,000 compounds had already been added to the Joint European Compound Collection, and we were expecting to meet the 200,000 target that the project had. And once that target is met, the portal is expected to be closed, and no new submissions would be allowed. So a few more details of this. The public compounds, that is the ones that have come in through this route, the library design side, as opposed to the FPA company compounds. These were, our ones, as you might think of them, were slightly larger in terms of molecular weight, but were slightly lower log P than the FPA contributed ones, but in the same overall ballpark. The public ones that we handled were a bit more 3D in nature, which was one of the key design objectives, and that was measured by fraction of sp3 and the PMI. The public ones, they explored a different chemical space to the FPA ones, which again was one of the design criteria. We specifically wanted compounds that were different in chemical space, and that was part of the selection process. So it was good that we achieved that. And when it came down to users selecting these compounds as the results of the screening hits, they were selecting the compounds anonymously, not knowing what the source was. But when you looked at that retrospectively, you found that the compounds from the libraries were about as likely to be chosen as the ones that were supplied by the FPA companies. So the compounds were generally considered to be suitable and tractable for follow-up. And the biological activity side of things is yet really to be fully explored, obviously, because a lot of that work is ongoing. But it's key that the biological activity profile of the public compounds is different to the FPA compounds, which again is what we would have hoped for. So, lessons. Overall, the project was a great success, but you always learn from what you do. So one of the key things, the key lesson is that the automation of this process of providing people with a web-based, relatively simple-to-use system that allowed you to put in these proposals for consideration, and then the similar systems for assessing them and deciding which libraries to take forward, providing that sort of automation had a huge payback here. The project just simply couldn't have happened if this was being done through a series of Excel spreadsheets or something like that. And so that automation really, it was critical to the operation of the library selection committee and the success of the project. Second point is about usability. Now, obviously, we all are very keen about providing very high usability systems here. And I think we did a reasonable job here. Most users managed to use the system okay, but putting in good usability does require hard work. It doesn't come for free. You have to resource it. And the project probably didn't have enough resourcing to be able to provide the real level of usability that I'd really have liked to do. I think we did a reasonable job here, quite a good job maybe, but there could have been more we could have done there had the project had a bit more resource. Yeah, one of the key things is we overlooked the management aspects of the system. What happens when something goes wrong? What happens when someone submits a buggy structure which causes calculations to go wrong or something like that? And all too often, when something like that happened, we were having to go in and manually fix the systems, where what we should have done is put in more effort up front, so we had built in better management capabilities into the system, so we could have handled those sort of things directly using the administrative interface, rather than have to go in and look in the database and look in the log files and things like that. So for these sort of long-running projects, you definitely need to invest quite a lot, and quite a lot more than you might think, in things like management and error handling functions. And the final lesson, which was in a way not unexpected, and actually John Overington commented on this at the beginning of yesterday, the crowdsourcing side of it really was pretty unsuccessful. We did have some libraries that were submitted by consortium partners, but the number was really very few. The vast majority of the stuff was come by the CROs and the academics who are part of the program. How we solve this, I don't know. It's still open, actually. If you've got some library design ideas and you want to earn a buck, you can still go to the system and register your ideas, and you can earn up to 5,000 euros for a good proposal. But just that cash reward on itself wasn't enough, really, to get wide-scale uptake. So, finally, thank you. Thank you for listening, and thank you particularly for these people who are heavily involved with the work. On the left-hand side, left column are the people who were mostly involved with the development. In the middle are the people from ChemAxon. I particularly want to point out Gabor Imri for the MadFast search and Tamas Pap for doing the IT and all the systems admin. They played a big part in this project. And people on the right were people from the ELF project itself, who played a significant role in these aspects of the project. So, as I said, the system is still running, and it will run for at least part of this year. If you want to try it out, you can. Here you are. You can even submit libraries, but stocks won't last forever. So get on there soon if you want to. Okay, so I think that's it. Thank you.